Well, welcome or welcome back, everyone. We seem to have a much more successful launching of the seminar today, so welcome. We acknowledge the traditional owners of the land in which this event is taking place, the land of the Wurundjeri people of the Kulin Nation, and pay respects to their elders and families past and present. So I'd really like to take this opportunity to welcome Dr. Lu Wen to the VCCC. Lu's been visiting us for the last 2 days. Dr. Wen was initially trained as a medical doctor in the School of Medicine in the Central South University in China and has obtained a master's degree in medicine in neurosurgery. His initial interest in research came from the Human Genome Project, and he received a PhD at the National Laboratory of Medical Genetics in Central South University. He then worked on zebrafish genetics and development, and as a postdoc in the School of Life Sciences in Peking University. He joined the Biodynamics Optical Imaging Centre, which we will hear some of his work from that centre in his seminar, I'm sure, at Peking as a research scientist in 2011. So his interests lie in developing and applying cutting-edge genomic technology, particularly to address fundamental and compelling biological issues related to medical problems. So welcome, Dr. Wen, and we look forward to hearing from you. Thank you.
So thanks for Melissa's introduction. So this is my first time being in Australia. So I find both the weather and the people very nice. So thank you. It was my great pleasure to talk about our recent work on novel techniques for epigenome profiling of single cell and their circulating cell-free DNA. So the epigenomics, including their DNA methylation, their histone modification, and their chromatin state, play crucial roles at both the development and the human diseases such as cancer. So we are interested in the dynamic epigenome during the development and human diseases, and we are developing our tools that can explore the areas that I was not able to explore before. So my talk consists of 3 parts. In the first part, I'll briefly introduce our work on the whole genome single base resolution analysis of hydroxymethylcytosine and in human brain. And in the second part, I will introduce the single cell multi-omic sequencing in early development and human cancer. And the third part, I'll introduce a novel technique for genome scale detection of hypermassive CpG islands in the circulating cell-free DNA. So DNA methylation of the cytosines in the CpG context mostly, it has long been thought as the only DNA modification in the human genome until 2009. It was found that the DNA methylation 5-MC can be oxidized by tagged proteins into hydroxymethylcytosine and then fructocytosine and carboxycytosine, and which represent active DNA demethylation pathway. So hydroxycytosine is of particular interest because it is relatively abundant in the human genome, especially in the brain. It's about 10% of the methylcytosine, and it's several times higher compared with other tissues. So the exactly of HMC in human brain is currently not known, and among many efforts to address these questions, we asked the genomic distribution of these new DNA modifications in the human brain. So we used a method called TAPSIC, was established by Professor He Chuan in Chicago University, that can give us a whole genome single base resolution of the hydroxycytosine map, and we also do the bisulfite sequencing that can give us a total DNA modification. including MC and HMC, then we could get a whole picture of the DNA modification in the human brain. So this is the map like. Actually, in the human brain, there's a baseline level of hydroxytyrosine about 20%. About 20%, even at the repeated sequences, you have 20% hydroxytyrosine. Except the major site, right? It has very low hydroxytyrosine and high methyl-cytosine. So at a single base level, you can find that hydroxytyrosine are very high at some special sequences like the CPG upstream CPG island shore of the TET promoters, and also at the poised and active enhancers. The two unexpected finding of this our study is that one is that the strain biased HMC and MC distribution in the gene body. If you profile the hydroxytyrosine and the methyl-cytosine across the gene bodies of red one is highly expressed, and the green one is moderately expressed, and blue is lowly expressed, and black is no expression genes. You can find that the hydroxytyrosine has the level increased in the highly expressed genes. We found that by the single base resolution map, that the hydroxytyrosine is actually not equally distributed on the sense strand, the solid line, and any sense, that the dotted line. It has high level on the sense strand in comparison with the antisense strand. Also, it's worthwhile for this is transcription correlated because this is high. The difference is most prominent on the most highly expressed genes, and this is worthwhile for the methyl-cytosine. Another finding is that the hydroxytyrosine has a peak at the exon-intron boundary regions, and in contrast to the gene body map, this peak is not transcription correlated. It occurs on both the I think you can point this. Both the gene that expressed highly expressed and the gene that do not expressed. So this finding suggests that there's a mechanical link between the demethylation and the elongation and the splicing. So the second part, I will introduce our some techniques that and our work on the single cell sequencing field. The single cell sequencing technology developed very fast during these years, and we applied this technique in two fields. One is the early embryonic development, in which the cell number is very limited. When one zygote meets one oocyte, then the cell from 1 divided into 2 and 4 and 8. So we must use the single cell sequencing technique to get a picture of this epigenome picture of these cells. And the second is cancer, in which the heterogeneity is a very important issue that is highly associated with both the outcome and the therapy of this disease. We have used the single-cell RNA-seq method that analyzes the early human embryonic development, and we found that the transcriptome of early development is very dynamic. That is different from from zygote to the from oocyte zygote to the 2-cell, 4-cell, 8 cells, very dynamic, and actually you can see the development of these cells just by the transcriptome. And we also developed novel single-cell RNA-seq techniques, including super-seq, that can detect both the poly-A plus and poly-A minus RNAs, and we have used this technique to analyze the circular RNAs during human and mouse early development. We have also established single-cell DNA methylome sequencing techniques, that is single-cell RBS. We have used this technique for analysis of dynamic DNA methylome changes during early human and mouse development, and identify the differential method region. Recently, we have developed two single-cell multiple omics sequencing techniques. One is a single-cell QSeq that is able to detect the chromatin state, DNA methylation, the copy number variation, and promote in individual cells. Another is single-cell tri-seq that is able to detect transcriptome DNA methylation copy number variation in an individual cell. So the single-cell QSeq is a combination of the NOMI-seq, as a nucleosome occupants and the methylome sequencing, and PIVAT as a post-Vasaf adaptive sequencing, allow the NOMI-seq to work at a single-cell level. And we also can detect the copy number variation of polarity in this assay. So NOMI-seq uses a GPC matrix that is able to measure the GPC site, the cytosine in the context of GPC site in the open chromatins. So, however, in the chromatin that was protected by the nucleosome, they are not able to measure this site. So, as the right panel, you can see we validate this method using the using the mouse birth stem cells. So the top is the black line, that is the naked gDNA. So actually, they're totally not protected by nucleosomes. So you have a very high level of GPC matrix. So for the bulk mouse ES cells, actually the most regions are protected by the nucleosome. So you can see that at the TSS region, there's a peak or the increase of GPC methylation level. So this promoter region is because there's no nucleosome at TSS regions. So we tested at the single cells by different color of line. Single cell has similar results with the bulk cells. So it means this technique can work at a single cell level. So we used this method to analyze early human early development, and we found that just by the left side is the t-SNE analysis. Just use the chromatin states, you can separate the cells of early embryo development. So there are different dynamic changes during the early mouse embryo development. So one finding of this study is that the chromatin open very fast at its regions. For example, you see the oocyte region at the left top bottom panel. The oocyte promoter actually is relatively closed, and the sperm is at the bottom. The blue one at the bottom is very close, compacted by the nucleosomes. So actually, quickly after fertilization, you can see increase of GPC level at the promoter regions. So actually, very quickly after the fertilization, the promoter began to open at the promoter at the regions. So also the right side is the all genes. You can see the heat map around the TSS profile. More red is the GPC level is higher. So you can see the sperm, of course, the sperm has no message level, very low level, because they're very compacted and at the oocyte. And so of course, you can see that the promoter region begin to more and more open. So we are also established a method as single-cell trial-omics sequencing method that was able to detect the epigenome or the genome, epigenome and transcriptome in individual cells. So the cancer is highly heterogeneous. So in both all of these levels, the epigenome heterogeneity, the genome generality, and the transcriptome heterogeneity, and we wish to explore them in individual cell. So this method, the principles of this method actually not complex. So after you lysis the cells after lysis cells, you spin down the nucleoside at the bottom of the tube. Then you get one of this half of this superlin out to analysis. Use the single cell RNA-seq analysis to get the single cell transcriptome. And then you can also analyze the DNAs at the bottom of the tube to perform single-cell DMSLOM analysis. So we compare the single-cell TRASIC with the single-cell RNA-seq alone and the single-cell RBS. We found that the single-cell TRASIC, the left panel, you see the right panel. Right panel, the single-cell RNA-seq is similar. You got similar number of detected genes with the single-cell TRASIC and correlated. So also for the single-cell RBS, you can see the detected methylation level and the correlation between the single-cell RBS and the single-cell TRASIC is high. We also found that we are able to detect the copy number variation by using the single-cell TRASIC data and also this is the HepG2 cells. You can see at the bulk level. Use the this is the middle panel. The uppermost is the SNP array result, and also the bulk W. The whole genome-wide bisulfite sequencing result from the previous published, and also bulk genomic DNA, and to show the copy number variation of these cell lines. Also, if you use bulk RBS and also single-cell TRASIC, we can also detect the same copy number variations of the HepG2 cells. So this means that the single-cell TRASIC can also detect copy number variations. One finding of this is that in the HepG2 cells, if you have a copy number variation, a copy number gain at the region, for example, the chromosome 2, you have a copy number gain, the green one, copy number gain. We will also have a copy number increase of the gene expression at these regions. So in the individual cells, so this means that if you have a copy number gain at some regions, the gene expression at this region will also increase. So we zoom in this chromosome 14 to 18. So you can see at when there's a copy number gain after 14 regions and 16 chromosome 16. So you will also have increased RNA expression at this region. However, the DNA methylation at this region is similar between the copy number gain and the no copy number variation region. We analyzed the liver cancer patient. So we analyzed the 25 cells at this region, and we are fortunate to find that they are composed of two subpopulations. And one is 7 cells, that is a small populations. Another is a big population composed of 18 cells. So if you just analyze the bulk, HCC bulk, you can see the HCC bulk is similar to the result of HCC bulk. It's similar to the big populations. So also as in the HepG2 cells, if you have the copy number gain at the regions, you will also have increased gene expression in these individual cells. Also, we find that the heterogeneity, DNA methylation heterogeneity is high in the primary tumors, HCC tumors, in comparison with the HepG2 cells. So there's indeed the high degree of DNA methylation heterogeneity in the primary tumor cells. We found that just by using the DNA methylation alone, we can also separate these cells into exactly the same two subpopulations as the copy number variations. And also the bulk is similar to the large population. So comparing these two populations, we can find there's some differentially methylated CpG island between these two populations. Also, if you had higher methylation level at the genes, at the S100 genes, at the promoter regions, if you have higher demethylation, and you will have low gene expression of these genes. Also, if we use when we use the gene expression data alone, transcription data alone, we can also separate the cells into the same populations. By comparing these two populations, we also find some differentially methylated genes, differentially expressed genes. So next, we have used the single-cell triseq to do more comprehensive analysis for colon cancers. This patient has metastasis. This is a grade 4 cancer, has metastasis and the lymphonoid and the liver metastasis. Also, we analyze the primary tumors and the lymphonoid metastasis, and also the liver metastasis. Several hundred cells for each site. And also there's a post-treatment metastasis, liver metastasis. This metastasis site is too small. It's invisible at the diagnosis, at time of diagnosis of this patient. So only after several months after the first operation, and the doctor found that there's a second metastasis site began to grow up. So the patient have received another operations of this site. So we are able to get the cells of this second metastasis site. So if by the transcriptome node, the individual cells can be classified into, most of the cells can be classified into type 2 the colon cancers by transcriptome. So as I've said before, so we can detect the copy number variation by the DNA methylation, genome DNA methylation date. So you can see this is a different site of the copy number variation of all the chromosomes, all the chromosomes at the different site of the tumor. You see primary tumors, we have four sites. Four sites, the site have different copy number variations. So those lymphocytes have different copy number variations. The metastases will cause different sites. You can see the copy number variations of these cells. Actually, we can draw a cell lineage map of these cells, of all these individual cells by the copy number variations. Because if you see a copy number variation at the mother cells, you can see the copy number variation of the mother cells, and at the basis of these copy number variations, if the cells have additional copy number variations, this should be the daughter cell of the mother cells. So we can actually to just deduce the cell lineage relationship between these cells. In the primary tumors, there are two populations, big populations. One is A and another is B. So the A populations, A0, has derived several daughter populations, and also the B have derived several two daughter populations. Actually, the lymph node metastasis is from the A population, A0. So A0 comes out. A0 population comes out of the primary tumors, and he derived 4 additional subpopulations. And also the liver metastasis is also come from the A0, and also shares a population with the lymph node metastasis. So it's possible that they also come from the indirect from the lymph node. Also, the post treatment metastasis is also come from A0 population, and it's give another subpopulation, A8 populations. So we can find the draw cell lineage, trace the cell lineage of this tumor. So actually, since we are doing tri-seq, we can compare the demethylation and the transcriptome of this A population and B populations. So what we found that is, you know that DNA, the cancer has a global hypermethylation and of course focal hypermethylation of the CpG island. So you can see that the non-NC means non-cancer cells, adjacent cells, NC has higher methylation, and the A populations, top panel, A populations, both A and B populations have lower methylation level in comparison with the adjacent tissues. So surprising later at the beginning, we found that the B population have even more lower methylation in comparison with A populations. So they are more abnormal than the A populations in the from the DNA hypermethylation side. So you know that actually A population comes out, but B population have not come out. So we compare the transcription of the A population and the B populations. We found that the B populations express more viral antiviral genes and immunogenes and anti the virus. We also find that they actually express more repeat sequences. So actually, it seems that because of the more abnormal DNA hypermethylation, the repeated sequence has been activated in more extensive way than in the B population, and because they are activated in a high level, they have triggered the viral antiviral action of the cells, so anti-fragment cell. So they maybe that cause that they are not easy to come out, and difficult to come out. So in the last part, I will introduce a novel technique for genome scale detection of the hypermethylation CpG island in the circulating cell-free DNA. So you know, the focal hypermethylation of CpG island is also a hallmark of human cancers. So in most tissues, the CpG island are not methylated. However, in cancer, the CpG island are highly methylated. These have been found a very long time. So our question is whether we can perform, the tumors can release the tumor DNAs into the blood as the circulating tumor DNAs. So this has also been found for a long time. And our question is whether we can perform genome scale detection of the hypermethylation CpG island in the circulating cell-free DNA. This is a technical challenge, actually technical challenge, because that the circulating cell-feeding tumor DNA is the amount is very low, also in a very low fraction, about lower than 1% in the fraction of the cell-free DNA that comes from white blood cells. Also, they're highly fragmented as a 160 base pair average, so it's one nucleoside. So they are both highly fragmented and in low amounts. So the methylation genome scale methylation method, including methylation rate, so you have used a lot. So they're excellent for tissue samples, and also RNA-based excellent for tissue samples. They are not able to work for cell-free DNA. And RBS use the site selection strategy. They're not for cannot be used for cell-free DNA that already fragmented. So we have an established method called MAXIC. It's a method CpG tandem amplification and sequencing method for detection. So this method is based on the finding that the CpG tandons, that is like CG, CG, CGG, is about 13,000 CG, CG, CGG in the human genome, and 80% of them are within the CpG island. So we first do the bisulfite treatment to convert the CG, CG, CG in the demethyled CpG island in the normal tissues into the UG, so they cannot be amplified. Then we use the random primer A to amplify, do the first amplification. Then we add the primer B that with the CG, CG, CGG at its 3 end for the second enrichment amplification. Then we added adapter for further amplification. So we first test this method in the fully methylated genomic DNA. You use this MSS1 to treat the genome, make all the CpGs are methylated. So we find that the MAXIC can detect about 9,000 CpG islands. Is about 1/3 of all the CpG islands in the human genome, and it also can detect one and more HepG2 3,000 CpG islands in the HepG2 and white blood cells. So the analytic sensitivity is very important for the cell-free DNA detection. So next, we tested the analytic sensitivity. We spiked the 40 methyl genomic DNA into the white blood cells in a fraction of from 100% to 10, 5 to 1, down to 0.25%. So we found that the MAXIC can detect the lowest fraction. And also we want to see the absolute detection limit of the DNA of this method, that we diluted the DNA from 100 and from 15 from 150 kilogram and down to the 7.5 kilogram. So it's about 1 to 2 cells, equal to 1 to 2 cells geometry. So this experiments tell us that the MAXIC is very sensitive, that can be used. So this is what the picture is like. We'll get the sequencing pictures. In the one meeting CpG island, there are actually 2 CG, CG, CG. And one is the red one, red triangle, as a forward direction. Another is the reverse one and the reverse reaction. So so you can see that actually all the reads are come from these 2 CG, CG, CG sites, and they're a very small at the point, error point. It's a very small fragment at a 13 base pair can be detected. So as a dilution range is very good. So then we next move to the tissue samples. we have tested the liver cancer tissues and non-cancerous adjacent non-cancerous tissues. By clustering, you can see the red point is the liver cancer tissues, and the green point is non-cancerous adjacent non-cancer tissue, and the green point is 3 normal liver tissues. So XSEQ can clearly separate most of the liver cancers from cancer tissue from adjacent lung cancer tissues. Also, we compare these two. We find the tissue differential message CpG island. Then we go to the plasma samples. We found that a little bit surprising at first sight that the liver cancers, the plasma of liver cancers has very prominently more hypermassive CpG island in comparison with the cirrhosis and normal individuals, non-cancerous individuals. So liver cancers can be very large. So this means that especially in the large states, they release a lot of tumor DNA into blood. We also find the plasma differentiated message CpG island. So we focus on the smaller tumors that is less than 3 centimeters. So yes, we have used the ORC area to find the most performance CpG islands. And we are also a little bit surprising to find that only half of the plasma differentiated message CpG island overlap with the tissue message CpG island. So, and it's like the Wilmington locus, so it's highly masked in the plasma, the red one, plasma of liver cancer patient, and in comparison with cirrhosis and the normal cancer patient, and also it's highly masked in the liver tissues, liver cancer tissues in comparison adjacent non-liver tissues. So we also found another set that we call type 2 that is only like the Shank2 locus here. We find that it is highly masked in the plasma of HCC patient and in comparison with cirrhosis normal patient. However, there's no difference between the maceration between the cancer tissues and adjacent and normal tissues. So exactly there are tissue-specific message sites that have, you can see red, they have highly masked. The type 2 one is low panel. They have highly masked in both the cancer tissues and adjacent non-cancer tissues. However, the low masked in water bottle cells. So they are actually the tissue-specific message site. In 9 patients, we have got both the tissue samples, adjacent lung cancer tissue samples, and the plasma samples. So we are able to, we ask a question, where did the abnormal DNAs in the plasma come from? Whether it indeed come from or come from the cancer tissues. So we compare you calculate the correlation of the markers between the plasma at the red bar, the plasma and the cancer tissues, and the green bar is the plasma and adjacent tissues, non-cancer tissues, and gray one is the tissue, is the cancer and adjacent tissue. So actually, we found that the correlation is high in 5 of the 9 patients, and this means that the indeed dominant mainly come from the cancer tissues. However, we found that the correlation is low in 4 patients, and the correlation, in contrast, the correlation is high between the plasma and the adjacent lung cancer tissues. So this actually had puzzled us for some time. And when we rank the samples, we found that all these patients have small cancers. So it seems makes sense that when the cancer is small, the release the when they release a lateral DNA into the blood, the lung cancer's the livers may will be damaged because of some reason we don't know, that release maybe inflammation, release more DNAs dominant into the blood. So this is a whole picture of the cancers. Also in this patient 44, we have also the post-surgery samples. That is the rightist panel. Actually, after the cancer was removed, the signals quickly go down. So this means that the non-cancerous relief of self-DNA, although they're also highly associated with the cancer. So in this patient, AFP is the most commonly used for detection of liver cancer. And AFP detected only in 2/3 of these cancers, and 1/3 of the cancers are not detected. However, all these patients can be detected by detectable by the self-free DNA. So this means the self-free DNA may be used for clinical diagnosis. In addition to this, recently we'll also go on to the colon cancer detections. So at this stage, we found that the colon cancer, of course, release less DNAs in comparison with the liver, especially the liver cancer could be very large cancer. So we found that when you use the CG site and a single base level methylation level, we can have more sensitive detection. So now we use the CG as a unit for our calculation. And we also find that differential method CG site. Also, we found that in this case, we found that the colon cancer, the release of the lung adjacent tissues cannot be found. So it's special for liver cancer. We also make a calculation by cross-validation using the tissue date. We first selected the methylated site as most highly expressed tissues and have low background in the independent set of normal tissues, and we calculate the bioscore by cross-validation, and we find if you have more markers, and the performance will be better at 60 markers. This markers is a CG site. If a 60 CG site detected, the performance became plain. And it just calculate the total positive markers. The number is similar to the random forest calculation. So we selected the 60 markers, make cross calculation, and we have analysis stage 1, stage 2 in the training group now. Now we are doing testing this. We can got ORC of 0.8 for the stage 1. So we also found that the cancer tissues the cancer livers are different from the colon cancer livers. The liver cancers are different from the colon cancers. So this right panel is the tissue result. You can see you use the low background markers that distinguish very clearly. And also the left panel is the plasma result. If you calculate the number of positive panels, we can actually distinguish the liver cancer from the colon cancers in blood at a high, at low error rate. So I'm in the fructose group, and we also, this is also of course a teamwork. It's a lot, many work, and also we cooperate the hospitals and also other people, and also just many students involved with this. So thanks. I'll take questions. Sorry, that's my point. And this.